conversation presentation is also of I see a lot of discussions around it. It's around sleep within the chaos community. I know for our household, sleep is it's something that we envy other people getting. And Dr. Lipton from Boston Children's Hospital is going to speak to the intersection of KS with the circadian clock. So excited to have this next presentation. Again, when we see this so often communicated, we know that there's a burden of care correlation with our children and even adults. There's also the kind of the precursor for regression around sleep. So, Dr. Lipton, when you're ready, I will turn the floor over to you.
Hello. Can you see my screen?
We can see you.
You can't see my screen. One second. Let's see. How about now?
Yes.
Great.
Perfect.
Okay. Well, thank you very much for including me and inviting me to speak. I'm Jonathan Lipton. I'm new to the Kleefstra syndrome community, so I appreciate the chance to be here. I'm a pediatric neurologist at Boston Children's Hospital, and I'm also a sleep specialist. I also run a basic science laboratory that focuses on the basic mechanisms through which circadian rhythms intersect with neurological disease on a variety of different levels. I'm going to give I wasn't exactly sure about the audience, so I'm going to give sort of a broad overview and then show you a little bit of data and describe actually an exciting collaboration that I think led me to this conference in the first place. So so I'll dive right in. So as I've heard from several of the talks that I was able to attend already, and I think from patients and from my colleagues, I think, and I'm guessing that for many of you, this is not a huge surprise that sleep dysfunction is a common bedfellow of neurodevelopmental disorders. This is the reason I got into this field as a pediatric neurologist. Sleep dysfunction is incredibly common across a wide range of neurological disorders, and especially in children with intellectual disabilities, autism, and other neurodevelopmental concerns. Most of the data on this comes from patient report, and so is largely dependent on questionnaire-based studies and subjective data. There's not a lot known about the actually underlying mechanisms, but I just made a list here of some of the sort of players, some of the disorders that where we see sleep dysfunction very commonly, and they include disorders like Fragile X syndrome, tuberous sclerosis complex, Prader-Willi syndrome, Dravet syndrome, and this list is by no means comprehensive. The list is actually probably extraordinarily extensive. And I guess the question for the talk today is, where does Kleefstra syndrome lie, and why and what could some of the mechanisms and explanations be? And so the short answer is we don't know yet, and that's part of the reason we're trying to engage in research on this. And one thing I wanted to say is we would really, I think, value input from the community. We'd love to hear more about what kind of dysfunction you see and what's important to you. The most common manifestations of sleep dysfunction in general is insomnia, which is the difficulty either falling asleep or staying asleep. In the few pieces of reports that are in the literature about specific to Kleefstra syndrome, what's reported is often frequent nocturnal and early morning wakefulness. And this suggests a degree of what we call sleep fragmentation or disruption of the sleep cycle. We wonder about things like sleep-associated epilepsy. And then when I hear about sleep fragmentation and difficulty with waking during the day and during the night, I often worry about circadian rhythm disruption. And we'll talk about that in a moment. Now, sleep is important. We often think of sleep as, we sort of unplug the computer and lie down at night. But there's indisputable evidence over the past 5 to 10 years that sleep has a completely fundamental role in our health, and especially in the health of children. So sleep dysfunction is associated with a whole host of health-related concerns, including, and those that I think that are relevant to KS patients, behavioral dyscontrol, a lowered threshold for seizures, mood disruption, metabolic disease, and an overall diminished quality of life, both for the patient and certainly for the family, especially if they're young children or children with neurodevelopmental challenges. And then, of course, there's these reports of these sort of dramatic changes in sleep, often associated with young adulthood or adolescence, followed by these psychotic-like or regressive-like episodes that I think still need to be further characterized. I know Zoe touched on it briefly, but these kinds of reports were certainly raised our ears and in part led to some of the early work that I'm going to show you. So before we move forward, and I'm going to tell you a little more about some of the mechanisms we think might be involved, I think it's important to have some working definitions. So we talk about sleep all the time. I think all of us know about sleep because all of us do it to a varying degree. But what is sleep from a scientific and clinical standpoint? So what sleep is sleep can be defined as a state of diminished consciousness. And the most important part of that first sentence is the second one, which is that it's reversible. So unlike more severe forms of diminished consciousness, what happens with sleep is miraculously we wake up and we're still the same person. We still have our memories often. Sometimes they're even improved by sleep. The other thing that's interesting from a scientific standpoint is sleep is species dependent, so different species sleep differently in different positions and for different amounts. And sleep is homeostatic, which we'll touch on a little bit more in a moment. Homeostatic means that sleep is always being regulated by a homeostatic mechanism, which means that the more you're awake, the more you need to sleep. When you just wake up from sleep, you should feel refreshed. And then this drive to sleep then builds up. And I'll review this again in a moment. And then one thing I thought might be interesting to the community from a research standpoint is that there is not a single animal that's been studied so far that doesn't have sleep or a sleep-like state, including worms, flies, mammals, fish. Sleep seems to be something fundamental to animal nervous systems. So it's really important. And of course, sleep has captured the imagination, well, certainly for myself, but it's captured the imagination of artists, poets, religious thinkers, etc. It's really still remains one of the great mysteries of human behavior and animal behavior. Okay, so when we talk about sleep, sleep is regulated by two main processes. I mean, it's regulated by many things, but we can break it down into two main processes, which I'm going to describe now. One is this blue line. It's a little fuzzy, but the blue line here represents the drive to sleep. So here in the middle of the night, your drive to sleep diminishes because you're sleeping. So as you sleep, you diminish this drive. Then early in the morning, you start over again and you build up the drive to sleep, and then you discharge it again the following night. Now, working partially with and partially against that drive is a circadian clock, which is really a clock mechanism, like a clock on the wall, that doesn't really depend on how much you've slept or whether you slept. It really depends only on your internal timing state. And I'm going to show you another example of that. This might be a little bit more intuitive. So if you imagine the drive to sleep as a flask, and we fill the flask with water, in this case, water is the amount of sleep you want. Over the day, you build up more and more of this water until it reaches some critical point, and then the flask has to pour the water out. So you pour the water out, you discharge your sleep drive, and then you start all over again, okay? Now, that's a state-dependent process, meaning that it depends on what you're doing or what state your brain is in. The longer you've been awake, the more you drive the drive to sleep. That state then drives the change, and then starts over again. Now, in contrast, there's a clock mechanism that underlies the timing of all of this. And just like the clock on your wall, this clock mechanism doesn't care if you've been awake a lot, whether you, I won't say it doesn't care whether you've eaten, but let's just say it doesn't care whether you've eaten, whether you've slept, whether you're happy or sad. Just like the clock on the wall, when 3 o'clock comes, 3 o'clock comes, and in an hour it will be 4 o'clock, etc. We have that represented inside our bodies, and it regulates many, many things, including sleep. So, and the reason it's important to understand this a little bit is what we're trying to avoid is insomnia. We're trying to avoid when these processes don't work together or each of them don't work on their own. And really for what we're trying to do is trying to understand how the underlying mechanisms that contribute to KS might contribute to sleep dysfunction in cells and in the brain. So we're going to focus now for a moment on this clock mechanism, not because it's the only important mechanism, but because it's one of the most accessible mechanisms to study. And it's also the focus of much of my research and expertise. And why do we study this? Well, we live on a rotating planet, and the planet spins, obviously, creating a light-dark cycle. And to adapt to that light-dark cycle, all organisms have an underlying clock that represents that light-dark cycle inside it. So it predicts what's going to happen during the time of day. And it divides up our cellular function and our behavioral function into wake and sleep, the use of energy and the storage of energy, and then many other processes, which I'll review in a moment. Circadian rhythms actually impact all aspects of life, from behavior to physiology to our metabolism, and even the expression of certain genes and proteins in the cell. So it's really a global organizing system that connects us to the rotation of the Earth. And because of that, it's really multi-scaled. And what I mean by that is that it exists across all scales of biology, from population levels, for example, like social networks, to organismal behavior, like a sleep-wake cycle, to tissue function, like whether or not your liver is going to make enzymes at a certain time of day, or whether or not you're more attentive at a certain time of day, et cetera, all the way down to cellular and protein function. And we can study rhythms, circadian rhythms of about 24 hours at any of these levels and integrate them across mechanistic levels all the way up to behavioral and complex behavioral levels. I won't spend too much time on this slide, but just to review for a moment, in mammals, we have a central clock inside the brain called the SCN that is required to as a pacemaker for clocks throughout the body. And that pacemaker, it's almost like a conductor of an orchestra. It doesn't play for the orchestra, but it keeps the time for the orchestra. But then each cell, each group of cells in the brain and in the body has its instrument, and that instrument in this case is a clock. And those clocks exist and beat and predict the functional needs of the individual organs in relation to the overall needs of the organism. And this is a highly integrated, complex system. And the reason I'm telling you all this is because I think it's important to understand that when we're talking about sleep dysfunction and circadian rhythm dysfunction, we're really talking about a whole person, how that affects a whole person, okay? So you can look at, this is sort of a joke, but you can look at a human being or a patient through the lens of circadian rhythms. And I think it's crucial to do that. And we really have to start changing our approaches to integrate timing into our approaches to medical care. So how does this all work? Well, in every cell of your body, and don't worry about all the details, but in every cell of your body, there is a clock. And I don't mean there's like a little set of gears and cogs, but there's a set of molecules that function like gears and cogs. And those molecules generate something called a feedback loop, where they turn on, in this case, these two guys, turn on a group of molecules that then inhibit them. So you have an up and a down, and an up and a down. And this cycle takes 24 hours and is recurrent. And the description of this cycle was awarded the Nobel Prize in Medicine about 5 years ago, or 6 years ago. Okay, now I want you to just focus on this one. And it's easy to remember. This particular gene, it makes a protein called clock, which is easy to remember. And the reason that we're focusing on it is because I'm going to show you a little bit of data that is not published yet, it's still preliminary, that suggests that the mechanisms in KS may intersect with this protein. So that takes us to our collaboration. So this collaboration came literally from having coffee with Sid and talking about ways we could find to collaborate. And we formed this wonderful partnership that now includes three main parts. There's my laboratory, there's Sid's group, including Sid and Zoe, and there's the laboratory of Joe Kosowski, who's a colleague at Boston Children's and Brigham. And he's really an expert in biometric measurements in the field, including for sleep and circadian rhythms. And what we're trying to do is to really innovate a nuts to bolts approach to circadian rhythm and sleep disruption for Kleefstra syndrome, in which my laboratory is studying the underlying mechanism and cellular function. Sid and Zoe are studying natural history and patient genetics and all the things they described. I won't pretend to summarize it. And Dr. Kosowski, Joe's group, is actually helping us make measurements in the field so we can now coordinate how specific disruptions that are occurring on a genetic level affect the clock. Now, how can we do that? We can do that because, like I told you, clocks are in every cell. So what's amazing is we can take cells, we can put them in a dish, we can do a little special doctoring to them, and that doctoring allows us to see how the circadian clock expresses itself by making light, and we capture that light. And so what you're actually looking here, these squiggly lines, actually represent the output of the circadian rhythm in cells in a dish. And what you'll notice is that the gray line are cells that are control cells. We just treated them with a control system. When we use a special tool to inhibit the gene that's implicated in Kleefstra syndrome, EHMT1, we suppress the amplitude or the strength of the circadian clock. So you can see that here, and that's dose dependent. So the more of this inhibitor we add, the more we suppress this down. So you can see that's, and this is for two different inhibitors that we used against this gene. Now we also corroborated that by using a drug that blocks the function of EHMT1, and we saw the same thing, a suppression of amplitude. And that was exciting because it told us that, that EHMT1 might be affecting the function of this positive part of the circadian clock, and specifically the clock protein. And so without getting into all the details, I'm just going to show you this beautiful kaleidoscope picture here, which really represents a blown up, this is something called super resolution imaging, which sounds all fancy, but really what it is it's a really, really sophisticated microscope that allows us to look at the nucleus of the cell with incredible detail, in this case about, around the resolution of about 100 to 150 nanometers, so billionths of a meter, 150 billionths of a meter. And what we're peering into is the nucleus of the cell. And where you see the green, that's actually the HMT1 protein. And where you see the magenta, that's clock. And where they overlay, that's white. And you can see that they spatially overlay with each other, okay? And you can see that here just shown as the actual individual dots blown up. This is 280 nanometers. So really, really, really tiny. It's not quite molecular level, but it's getting close. So the other thing that we've done, and don't worry about what this means, when you see these two black lines here, what this is telling us biochemically is that these proteins, the EHMT1 and the clock protein, not only are they spatially connected, but biochemically they're talking to each other. So another really exciting and promising result. And to summarize what we can do with this is what we're trying to do now is we're really trying to take the clinical and translational information from the Kleefstra clinic, including genotype and phenotype information. We're recruiting patients so that we can take that information and now measure what's actually happening in the field with behavior and physiology, and then take when possible, take cells and samples from the patients and then model that behavior in the laboratory. So the goal is this three-part model with, of course, as much engagement from you guys as possible. So this is just an example. This is actually from the first patient that we measured, and we're still in the process of analyzing. We've now, I think, obtained data from 7 patients, but we're still analyzing. But I just wanted to show you that this is from wearing a device at home for a few weeks. We can actually get really high quality data here over almost 2 weeks that tell us about basics about sleep staging, heart rate variability, and actual activity. And from that, we can do our fancy analysis and measure things like how strong the rhythm is, how much it's changing in relation to the light-dark cycle, et cetera. And we're coupling that with cells from the lab that we can then doctor, work with, differentiate in different ways if we want to, measure the rhythms, and then, of course, correspond that with information from patients themselves about their sleep patterns and sleep concerns. So I want to thank my lab for participating in this work, especially Dr. Sarah Ferraro, who's a really dedicated and very talented postdoc in my lab, who's responsible for all the work that I showed you. She was accompanied by 2 talented undergraduates, Lyric and Tamar. And then I have to thank Sid and Zoe and Joe for amazing partnership and for starting on this exciting collaboration. None of it would have been possible without support and help from iDefine and from our colleagues and mentors at Boston Children's. And finally, I wanted to acknowledge the support of the Translational Neuroscience Center, who supported this work last year, and we actually just were awarded the renewal for funding for another year. So we're really excited to continue this and hopefully make some important progress. And I just wanted to acknowledge about a month ago, the beneficiary of the TNC passed away. So I just wanted to dedicate this talk to Mrs. Zander and acknowledge her support. Thank you so much.
Dr. Lipton, great talk. Incredible. I'm really excited about this particular study. I wanted to ask if you were able to share if we have any sort of recruitment goals for this study and who all the study is open to.
Yeah, so I think that question is best answered by Zoe, actually, but I will say that from I think that we're really getting off the ground now, and we really started to recruit from within the BCH clinic, and we've had I think a really nice response so far. I think our goal was to sort of do a first pass with these first 7 to 10 patients, but then once we kind of get our feet wet, and I think we're pretty close. We have talked about opening it up to the whole community, but maybe Zoe can address that. I see her coming in here.
I figured I'd bring her back.
Yeah, that's a good idea.
I'm back, baby.
Beyond my page.
I was just writing a response in the chat, but this is perfect. The enrollment is open to anybody who wishes to participate. So I can send a recruitment flyer, Jeff, for you to officially post on iDefine. Our recruitment goal is 50, and we just enrolled our 13th family. So we're well on our way, and it's been fantastic working with families. As I kind of alluded to in my talk, this is kind of a choose your own adventure protocol. So if you live far away and maybe your child isn't, amenable to wearing a Fitbit, you can participate by, doing the remote questionnaires about your behavior, your child's behavior and sleep. You can wear the Fitbit. Joe's team helps me ship them out, and Jeff, yours is about to go in the mail, so fantastic.
Yeah.
And then for those who are able to come to Boston, that's the easiest way to collect skin biopsies for the fibroblasts and the cellular studies that Dr. Lipton's lab is doing. So yeah, there's a lot of ways to participate, and any participation is welcome. We would love to have anyone who's open.
Yeah, I mean, I was going to offer and just be transparent. Anybody that has any questions about what this process likes, we're about to go through it. Like I said, I am all in on this study. I'm so beyond excited about it because I think it has ramification. Like you just alluded to it, Dr. Lipton. I mean, the role of sleep is so dramatic in the entire ecosystem, right? And so I think it feels very fundamental that we need to understand what's happening here and get as much data because it's going to inform so many other things downstream, right? So, yeah, I'm beyond excited. So I can't wait for Ella to get her Fitbit. I saw your email come across yesterday. So thank you for that. There's a couple of questions here that are popping into the Q&A. Are you looking at hormones in sleep?
We are not in this study yet. I think that's a very good question. Many hormones are under strong clock control. But then there are hormones that are actually specifically under sleep control. So there are hormones that require that you sleep. So for example, like growth hormone only peaks during slow wave sleep. But cortisone, for example, it demonstrates a circadian rhythm that is affected a little bit by sleep, but is mainly just driven by a timer, the internal timer. So it's a very complex issue. I think at this point, it's not the main focus, but it's definitely something that would be a ripe area of future investigation.
Are international families able to take part in the study?
At least, yes, in the context of the online questionnaires. I'd have to check with Joe's team about mailing Fitbits internationally just because of battery concerns. I'm not sure if the devices, I don't know about mailing, but at the very least, families are welcome to participate wherever they live, at least in the questionnaire component.
Right. Another question is the sleep data to which Dr. Lipton referred over a 14-day period derived from an actigraph or simply from a Fitbit?
It's an actigraph that's derived from a Fitbit. So the Fitbit actually is able to record heart rate, movement, and from the coupling of the heart rate and the movement, we can actually then make computer-based measurements that correlate well with sleep. And there's a separate study actually going on here at Boston Children's, a colleague of mine, where they're directly assaying exactly how well the Fitbit correlates with actually polysomnography, which is sort of the gold standard for measuring sleep. Preliminarily, I think that it's very encouraging, but the formal analysis of that is being done.
Okay. There's a question around the Inspire 3 Fitbit. I assume that's a version of the Fitbit. My knowledge of Fitbit is limited.
Yes, I've become familiar with different models of the Fitbit. So right now we're sending out Inspire 2s, but with our renewed funding, we'll be buying Inspire 3s. So yes, coming soon.
Amazing. There's two more questions and then we'll pause because again, this is like the last conversation where there's so much discussion within the community that I think we could have, a whole hour session of Q&A. But there's a question about the thoughts of melatonin usage.
Yeah, so I mean, again, as a sleep physician, I would need to talk to a patient about a specific situation. I mean, in general, melatonin is a safe over-the-counter supplement. The issue is, of course, is that it's completely unregulated and it's often misunderstood and misused. So, without getting into the weeds here, in general, melatonin is effective in children with autism and intellectual disability. I can't say anything specific about, Kleefstra syndrome, but in general, it's safe, but you have to get it from a reputable source and, use it properly. So,
yeah, I think that's one of those questions that's probably better answered in clinic one-on-one. It's a little bit it's a little bit tricky for probably both of y'all, right, to answer anything kind of unequivocally. Just making sure that there is a certain component of this that's very specific to the individual that we're talking about, and so I think understanding that a little bit better will help frame it up. And then the last question, Eric, has brought a really interesting point forward, and whether or not we know if sleep has ever been used as an endpoint in clinical trial, because as we talk about things that matter to the community, obviously
Yeah,
this is one of them. So have you ever had any
yeah.
evidence of this being used?
Yeah, so not in Kleefstra syndrome, obviously, but there are a number of studies and a number of efforts to do this. So it's a long answer. I won't give you the long version, but the short version is yes. There is a great interest, especially for certain disorders. The one place where it's been done the most, obviously, is in Angelman syndrome, with mixed success, I would say. But it's certainly being looked at in neurodegenerative disease, both from Alzheimer's, Parkinson's, et cetera. And there's a growing interest for doing it in developmental disorders. So that's a great question, and yes, I think there's a lot of interest in doing that. A lot.
Great. Well, thank you both. Like I said, I am really looking forward to personally being involved with this with Ella. If anybody from the community has any further questions about getting involved with the sleep study, it's amazing. I don't want to just blow by the point that this is being funded by the Rosamund Stone Zander Translational Neuroscience Center. Like, that's fantastic. That's part of the BCH hemisphere up there, right? That they're critical. Kira and Maya, who are longtime iDefine partners. I met Kira the first time I ever came to Boston, right a couple months after Ella's diagnosis, and so just seeing these partnerships continue to flourish. Eric had used the word halo effect about how these relationships are so kind of intertwined, and so that's exciting to hear. And obviously, however iDefine can support, this is one of those critical studies and points of need for the community. So we want to be involved as much as possible. So thank you both very much.
Thank
Thank you
you.
very much. Take care.